Modern medicine still relies on single signals. One blood marker, one scan, one symptom. But biology doesn't work in single signals. It works in networks. And right now, a quiet shift is underway. We're moving from reactive to predictive medicine, not because of one breakthrough, but because we can now read multiple layers of biology at the same time. This is the rise of multi-omics diagnostics, and it's already changing how we detect disease. Most diagnostic tools are built around a simple idea. Look for one thing that indicates one problem. But diseases don't start as single events. They start as subtle changes across DNA, proteins, metabolism, and even the microbiome, long before symptoms appear. By the time a tumor is visible on a scan or a blood marker crosses a threshold, the underlying biology has been shifting for years. The problem isn't that we don't have data. It's that we've been looking at it one layer at a time. Multi-omics changes that. It connects several layers of biological information: genomics, epigenomics, proteomics, metabolomics, and the microbiome. Each layer tells part of the story, but the real insight comes from the pattern across layers. Multi-omics isn't about collecting more data. It's about integrating data. Here's how it actually works in practice. It usually starts with a single blood draw. From that one sample, labs can extract fragments of DNA, chemical tags that show which genes are active or inactive, proteins in the blood, metabolites that reflect metabolic activity, and microbial DNA from the gut and other microbiomes. Each of these becomes a different data set. It's asking whether the overall pattern matches a known biological signature. Machine learning models look for patterns across layers, not within a single one. They identify combinations of signals that tend to appear together in the early stages of certain diseases. It's not asking whether one marker is high or low. It's asking whether the overall pattern matches a known biological signature. For example, the early pattern of a specific cancer, or the trajectory that precedes cardiovascular disease. The output isn't a diagnosis. It's a risk-weighted, biologically contextualized insight that clinicians interpret alongside imaging, symptoms, and medical history. This is what makes multi-omics powerful. It captures early, subtle shifts across multiple systems long before traditional tests detect anything unusual. One of the clearest real-world examples is early cancer detection. Tumors shed fragments of DNA into the bloodstream, but the DNA sequence alone doesn't reveal much. The real signal is in the methylation patterns, chemical tags that show which genes are switched on or off. When you combine methylation with fragmentomics and proteomics, you get a biological signature that can identify cancers earlier than imaging. These tests exist. They're being trialed. And they can catch cancers at stages where treatment is far more effective. This is multi-omics in action, not guessing, not predicting, detecting what was previously invisible. Another example is cardiovascular and metabolic risk. Proteins change long before symptoms appear. In cardiovascular disease, certain protein patterns can indicate risk years in advance. Not because one protein is elevated, but because dozens shift in a coordinated way. Multi-omic models can read these patterns and estimate risk with far greater accuracy than traditional markers. This moves medicine from "you had a heart attack" to "you're trending toward one, and here's how to intervene now." It's a shift from crisis response to early course correction. We see the same thing in chronic disease. Conditions like diabetes, IBD, and autoimmune disorders emerge from long-term interactions between the immune system, metabolism, and the microbiome. Multi-omics can detect early metabolic signatures that reflect these interactions. Not one marker, not one microbe. A pattern. This allows clinicians to identify risk before symptoms escalate, and to tailor interventions to the underlying biology, not just the visible outcome. This is why multi-omics matters. Instead of snapshots, we get trajectories. Instead of isolated markers, we get biological context. Instead of reacting to disease, we can anticipate it. It doesn't replace clinicians. It gives them a richer signal, one that reflects how the body actually works. And it's already reshaping how we detect cancer, predict cardiovascular events, and understand chronic disease. There are constraints. Multi-omics requires large, diverse datasets. Models must be validated across populations, not just in controlled trials. Interpretation must be clinically meaningful, not just statistically interesting. And because biological data is deeply personal, privacy and governance matter. But the direction is clear. For the first time, we can read the early signals of biology, not just the late-stage consequences. This is the shift from reactive medicine to predictive medicine, and it's one of the most important transformations happening in healthcare today. If you enjoy learning about the breakthroughs shaping our future, consider subscribing for more.